Welcome to Oddly Real. I'm Nem Stefanovic. Some of the most complex and fascinating structures and processes in the universe can be found right here on Earth as part of life. In particular, cells, the building blocks of life itself, contain a wide variety of these structures and processes. One such process is how energy is generated in cells using the electron transport chain, or ETC. This not only powers the cell, but it also powers the whole life form itself. That includes human beings. In this episode, we're going to explore the energy production process in our cells. Additionally, we're going to look at multi-protein structures in the cell, called complexes, which facilitate this operation. These are quite literally nano-scale machines with designated functions that are in continual operation, like you would imagine in any factory. We'll understand the inputs and the outputs of the ETC, including the reason why oxygen is so important to life, and the consequences when the ETC is disrupted. We'll also look at an amazing nano-scale motor that is used to manufacture ATP molecules, which is the fuel of the cell. Cells, in both animals and plants, possess specialized components or organelles that are called mitochondria. Mitochondria are considered the cell's powerhouse, where a molecule called ATP is produced. ATP molecules are able to capture and store large amounts of energy, which the cell can release and use to power its various operations. The ATP molecule acts like the fuel of the cell, and it is the main source of power to keep cells operational and alive. One cell will contain thousands of mitochondria, and each has various structures and components within it. However, we will be restricting our attention to just a few relevant parts related to certain energy harvesting activities. In particular, mitochondria contain an inner fluid called the mitochondrial matrix that is contained by an inner membrane layer. There is also an outer membrane that surrounds the inner membrane, which includes a space between called the intermembrane space. The components and the machinery in the form of protein complexes to produce ATP are embedded within the inner membrane. One part of this process is called electron transport, which involves releasing energy to pump protons out of the mitochondrial matrix. These protons are used to set up a proton gradient across the inner mitochondrial membrane, which will power the second part of the process. The second part of the process is called oxidative phosphorylation, which uses the proton gradient to power a protein complex called ATP synthase that manufactures the ATP molecules. Within the inner membrane, there are 4 different complexes labeled 1 through 4, which are large multi-protein structures, as well as 2 smaller intermediate transport molecules. This is what we call the electron transport chain, or ETC. These structures, along with ATP synthase, are ancient. They are used across animals and plant cells as well as bacterial cells. In bacteria, there are no mitochondria, so these mechanisms are embedded directly in their cell membranes. We said there were thousands of mitochondria, and each mitochondria has thousands of ETCs, so there are millions of ETC pathways in one cell. We will trace through each step as the electrons travel through the complexes along a path that is increasingly favorable to electron reception. If the cell acts like a little factory, then the protein complexes in the ETC act like nano-scale machines. Let's start with the inputs to the ETC, which are special charge-carrying molecules called NADH and FADH2. The cell has multiple different processes to extract energy from carbohydrates, amino acids, and fatty acids. We call these oxidative processes because they result in the removal of electrons from molecules. Some of these processes, like the citric acid cycle and fatty acid oxidation, take place within the mitochondrial matrix. The outputs of these processes involve the creation of charged molecules of NADH and FADH2, as well as a little bit of ATP. NADH and FADH2 serve as the inputs of the cell's power generation process to output much larger amounts of ATP needed to operate the cell. Think of them like little batteries that power the ETC complexes. The electron transport process begins at complex 1. NADH is delivered to the complex where an electron moves from NADH into the complex. Energy is released, which is used by the complex to pump protons from the mitochondrial matrix through the membrane and into the intermembrane space. This accomplishes two things. First, it increases the concentration of the protons in the intermembrane space. Second, it increases the charge in the intermembrane space relative to the charges in the matrix. This creates a charge difference across the membrane. This results in what we call an electrochemical gradient across the inner membrane. Why does the cell do this? We'll see shortly that electrochemical gradient is what is going to power the ATP production process later on. The second stage of the electron transport process happens via the coenzyme Q molecule, also called CoQ or ubiquinone. This is a smaller molecule that acts to transport electrons from complex 1 to complex 3. It accepts electrons in pairs from complex 1, but delivers them one at a time to complex 3. It looks like we skipped complex 2. What is its purpose? Indeed, it's not a pathway between complex 1 and 3. Instead, it acts as another input to the ETC, but not for electrons from NADH. Instead, it's a pathway for electrons from FADH2. Complex 2 accepts electrons from FADH2, which are then transported to complex 3. It uses the CoQ molecule, like complex 1, to facilitate the transportation. Keep in mind, there are thousands of these CoQ molecules, and any of them can be involved with this delivery. When the electrons make it to complex 3, once again, the multi-protein complex uses the energy to pump more protons into the intermembrane space. This further reinforces the electrochemical gradient across the membrane. The electron is then transferred to the next stage in the ETC, the Cytochrome C protein. The Cytochrome C protein plays a role as a transporter for the electrons in the ETC to move between complex 3 and complex 4. Its small size and water solubility make it highly mobile within the membrane to facilitate the electron transfer. This protein is found across species, being part of plant, animal, and bacterial cells. This protein also has a fascinating alternative use that is separate from the ETC. Cytochrome C can detect critical oxidative damage to a cell called cardiolipin peroxidation. This results in the release of the cytochrome C protein out of the inner membrane into the intermembrane space. This action further triggers a series of steps to cause cell apoptosis, which is a programmed cell death, like a cell suicide. It does this as a preventive measure when there is a significant amount of damage or issues to the cell that it might threaten other cells in the larger organism. Finally, the last stage of the ETC is the delivery of the electron to complex 4 from the cytochrome C protein. Once again, complex 4 will use the electron energy to pump more protons into the intermembrane space. The last place for the electron to go is into an O2 oxygen molecule receptor in the mitochondrial cell matrix. In fact, that's one of the main reasons why we need oxygen for life. It is oxygen that carries away electrons from the complex 4 stage of the ETC to free up the pathway for new electrons to be loaded for more energy extraction. The oxygen molecules in the mitochondrial matrix that possess these extra electrons take one more step in this process. A reaction occurs where the O2 molecule, along with 4 electrons that were removed from the ETC, can combine with 4 free protons to produce 2 molecules of water. If not enough oxygen is available, then the electrons from the complex 4 stage cannot be removed, causing a backup in the ETC. This would lead to a backup all through the chain back to the inputs, where NADH and FADH2 would not be able to use their electrons to power the other ETC complexes. This means the ETC could not create the electrochemical gradient to make ATP to power the cell. Clearly, oxygen is important to us. In fact, the poison cyanide binds to the complex 4 of the ETC, blocking that last step of electron transfer to oxygen. When this happens, even with small quantities of cyanide, the ETC backs up as described, leading to cell asphyxiation and death within minutes. It's worth noting that the ETC process is really a way to extract the energy provided in the form of electrons from the NADH and FADH2 molecules. The energy is used to trigger multiple proton pumping actions, the creation of an electrochemical gradient across the inner membrane, and some transport and recycling actions. Energy is carefully extracted at each stage and used very efficiently. This is different from the release of energy using combustion, where large amounts of energy are released almost all at once, with much of it lost as heat. Finally, the ETC creates the electrochemical gradient across the inner membrane in order to power another process called oxidative phosphorylation. This actually creates the ATP molecules that ultimately power the cell. In order to do this, the cell uses a truly amazing device called ATP synthase, or complex 5. This is quite literally a small nano-scale motor. It consists of 2 multi-protein units that are connected together through a rotor stalk called the gamma unit. The base of the stalk is housed within the inner membrane, and it's called F0. The head of the device at the other end of the stalk is called F1, and it is positioned within the mitochondrial matrix. The ATP synthase is powered by the high concentration of protons that were pumped into the intramembrane space by the ETC. There's an opening in F0 that allows the passage of the protons through it into the mitochondrial matrix. When a proton enters the passage, it loads the C ring in F0, triggering the rotation. Energy from the proton is used to turn the gamma unit in the ATP synthase. This turning action causes conformational changes in the head of F1, meaning that parts of the head change shape to manufacture the ATP molecules. The C ring is also linked to the gamma unit, so it too rotates. The gamma unit is able to conduct full 360-degree turns as it operates. For each full turn, 3 ATP molecules can be created, each one being formed from one of 3 ATP-making sites. Each of the ATP-making sites can be in one of 3 conformational states. Those states are loose, tight, and open. When in the loose state, ADP and a phosphate P bind in the site. Next, in the tight state, the substrates are clamped down on to form ATP. Finally, in the open state, the ATP is released back into the matrix. In fact, this process is repeated in that exact order, loose, tight, open, for every turn of the gamma rotor to make 3 ATP molecules per full rotation. What's the total energy output of this process? We can take this all the way back, even before the ETC and ATP synthase phases, to start from a single glucose molecule as a source of energy in the body. If we then traverse the cell's metabolic pathways, they produce the NADH and FADH2, which powers the ETC. The ETC creates the electrochemical gradient of protons to ultimately power the ATP synthase and produce ATP molecules. End to end, this yields somewhere between 27 and 38 ATP molecules per molecule of glucose. It is truly astonishing to see how the most fundamental unit of life, the cell, behaves in its power generation process. It's really not unlike a nanoscale manufacturing facility, complete with batteries, fuel sources, and even manufacturing equipment. Amazing. I hope you enjoyed this episode, and remember, oddly real, really interesting.